Hello, everyone. Thank you for joining today's session. Our topic is improving the analytical performance of PCR assay via nucleic acid extraction, with a focus on two widely used methods: spin column extraction and magnetic particle extraction. I'm Jessica, a scientist at CD Bioparticles, and I'll also introduce some of our specialized products that can support your laboratory workflows. Before we dive into extraction methods, let's briefly review why PCR itself is so important. Polymerase chain reaction, or PCR, has been a cornerstone of molecular biology since its invention by Dr. Kary Mullis in 1983. It enables exponential DNA amplification and has broad applications in diagnostics, forensics, and gene analysis. From identifying genetic fingerprints in forensic cases, to detecting infectious diseases such as COVID-19 and monkeypox, PCR is essential in both research and clinical laboratories. However, what often determines the reliability of a PCR assay is not just the amplification step, but the quality of the DNA or RNA template that we start with. This leads us directly to the importance of nucleic acid extraction, the very first step in any PCR workflow. The analytical performance of PCR strongly depends on the integrity and purity of nucleic acids. If inhibitors such as proteins, salts, or detergents are present, amplification efficiency drops, sensitivity is reduced, and false results may occur. That's why extraction is critical. It consists of two stages. First, cell lysis to release the nucleic acids, and second, purification to remove unwanted contaminants. Simply put, good PCR begins with good extraction. Now let's look at the different cell lysis strategies available, since this is where sample preparation begins. There are four main categories of cell lysis. Mechanical lysis, using devices like bead mills, provides high efficiency but can risk sample degradation. Physical disruption methods, such as thermal lysis or cavitation, are scalable but often costly. Enzymatic lysis is very specific but requires time and expensive reagents. Chemical lysis, using detergents or alkaline solutions, is simple but may leave chemical residues that interfere with PCR. Each method has trade-offs, and the choice depends on sample type, downstream application, and throughput needs. After lysis, we move into nucleic acid purification. Let's explore the main extraction approaches. Currently, three strategies are commonly used for DNA and RNA purification. Organic extraction, using phenol chloroform, is the traditional gold standard, but it's labor-intensive and hazardous. Spin column extraction, which relies on silica membranes, is widely adopted for its balance of simplicity and purity. And finally, magnetic particle extraction, which has become increasingly popular for its high throughput and automation compatibility. Today, we'll focus mainly on spin column and magnetic particle methods. Let's begin with organic extraction. Organic extraction has been considered the traditional, gold standard method for RNA purification. This method, often performed with reagents such as triazole, was first popularized in the late 1980s by Komczynski and Sachai. It relies on a monophasic solution of phenol and guanidinium isothiocyanate, followed by chloroform addition. After centrifugation, the aqueous phase contains RNA, while proteins and DNA remain in the organic and interphase layers. Organic extraction works with many sample types, including cultured cells, tissues, blood, and even plant material. It is highly reliable for removing proteins, and the denaturation occurs quickly, which stabilizes RNA immediately. However, there are limitations. The process is labor-intensive, low throughput, and not automation-friendly. Handling phenol and chloroform requires strict safety precautions, and managing chlorinated waste is a challenge for modern labs focused on greener solutions. Today, while still respected for its reliability, organic extraction has largely been replaced by faster, safer, and more scalable methods, namely, spin columns and magnetic beads. In spin column extraction, a silica membrane is used to bind nucleic acids under specific salt conditions. The workflow involves four main steps: lysis, binding, washing, and elution. Through repeated centrifugation, Impurities are removed while the RNA or DNA remains bound to the silica. This approach is versatile and works with nearly all sample types. Now, let's look at the strengths and weaknesses of spin column extraction. Spin columns provide high-purity nucleic acids, with ready-to-use kits that simplify laboratory operation. They are suitable for medium to high-throughput workflows, especially in diagnostic settings. However, there are also some limitations. Large sample inputs or viscous materials may clog the column, and centrifugation steps can be labor-intensive for large-scale automation. At CD Bioparticles, we provide optimized silica spin columns that help overcome these issues and provide consistent, reliable yields. Next, let's turn our attention to magnetic particle extraction, a more modern and automation-friendly method. In magnetic particle extraction, nucleic acids bind to the surface of functionalized magnetic beads. When an external magnetic field is applied, the beads are immobilized, allowing impurities to be washed away easily. Finally, purified DNA or RNA is eluted. This approach eliminates centrifugation and makes it especially well suited for automation. Let's compare the advantages and disadvantages of this method. Magnetic bead-based extraction is fast, typically requiring only 30 to 60 minutes. It produces very high-purity nucleic acids and requires only small sample volumes. Most importantly, it integrates seamlessly with automated instruments, enabling high-throughput processing. On the other hand, viscous samples may limit bead movement, and specialized equipment is sometimes required. At CD Bioparticles, we offer several solutions, including DNA and RNA magnetic silica particles, which provide excellent performance for both manual and automated systems. So how do spin columns and magnetic beads compare overall? Let's summarize. This table highlights the key differences. In terms of purity, both methods are excellent, but magnetic beads often deliver slightly higher consistency. For throughput and automation, magnetic particles are the clear choice. Spin columns, however, remain highly cost-effective and practical for routine laboratory use. Ultimately, the decision depends on your workflow scale and requirements. And this is where CD Bioparticles can provide tailored solutions for both approaches. Let's take a closer look at our product portfolio. For spin column workflows, we supply silica spin columns. For magnetic bead-based workflows, our flagship DNA and RNA magnetic silica particles offer uniform particle size, high binding capacity, and reliable yields. In addition, amino group or carboxyl group functionalized silica magnetic particles provide flexible surface chemistries for customized extractions. We also offer comprehensive DNA and RNA extraction and purification kits, and bulk purchasing options are available. Now, where can these solutions be applied? Let's look at real-world applications. Our extraction technologies support a wide range of applications. Viral nucleic acid detection, including COVID-19 and HPV. Clinical diagnostics and point-of-care testing. Food safety testing. Environmental monitoring. And, of course, academic and pharmaceutical R&D. These applications highlight the flexibility and reliability of our products in diverse settings. To further illustrate performance, let's review some supporting data. To summarize, PCR success begins with high-quality nucleic acid extraction. Both spin columns and magnetic beads are powerful tools, each with unique strengths. CD Bioparticles provides optimized solutions for both methods, tailored to your workflow needs. We invite you to connect with us to explore how our products can enhance your assays. Thank you for your attention, and we look forward to collaborating with you. For more information, please visit www.cd-bioparticles.com.